I can share my screen. Say sure.
Edouard, I think you have to unmute yourself.
Okay. Do you hear me? Is it good? Okay, perfect. So good afternoon, good evening, and good morning, everyone. Thank you for joining us today for this mini symposium from all the corners of the world. Fluicell AB and NPI Electronics have joined forces today to offer you a very innovative and interesting mini symposium. I am Edouard Janicki. I work for Fluicell AB, and I will be chairing this meeting today for you. Today's main topic will be to make you discover new perspective in single cell signaling by highlighting some of the latest work done in the area. Yes, so the program today will be a first talk by Professor Karsten Hoffmann from Jena University. The topic will be probing receptor function with intramolecular threat sensors. Then Dr. Matija Rožnik from Fluicell AB will be talking about microfluidic system for targeting individual cell. Then Professor Kent Jadmark from Karolinska Institute will join us for a couple of minutes to talk about the novel antibodies for treatment of pain. And then we will close this meeting by having our CTO, Dr. Gavin Jeffries, talking about patterning tissue for cell microenvironment engineering. But before that, let's start with, let's start with our partner and distributor that I have the pleasure to introduce, Dr. Jens Loser from NPI Electronics. So NPI Electronics is a company based in Tam, Germany. NPI is a manufacturer of electronic devices for life science that support scientists in designing and building recording setups, and that provide customized solutions for imaging and electrophysiology. Jens, hi. It's time for you to present maybe a little bit more what you are doing at NPI, and it's your turn to speak.
Yeah, thank you very much.
So you can start sharing.
Yeah. So it should be sharing now my screen, is it right?
So we don't see now your screen shared, but we might see it in a couple of seconds, maybe. Yes.
You should see it now?
Yes.
Right. Yeah, thank you very much, Edouard. I'm Jens from NPI Electronic. We are, as you already mentioned, a company that builds electronic instruments for now over 35 years, since 1984, and we are based in Hamm in Germany. We have always been in close relationship to science. For example, we are a member of the Bernstein Center for Computational Neuroscience in Munich, and also of the EU GLIA PhD consortium. This is what some or many of our products look like. You might have already seen these black devices with many knobs and switches on front panels. When you see those, you can be pretty sure it's NPI Electronic. Most of these devices are amplifiers for intra and extracellular recordings that we build. We also have temperature controllers, high voltage stimulators, and application systems for substances, either as pressurized systems or also as iontophoretic systems. Recently, we started building optical components, for example, light sources for optogenetics or fiber-based fluorometry. And all of these devices are developed and produced in TAM here in Germany. Some of our recent devices is, for example, this fiber optometer for fiber-based fluorometry. And we recently built and developed this amplifier. We removed all the knobs and switches from the front panel and instead have a software which controls the amplifier now. And the software is also capable of acquisition of data. What makes NPI's instruments so special is mainly their analog core, which they are all based on. And this guarantees low noise and superior performance. And the knobs on the front panel allow the experimenter to control and to fiddle on stuff while they do the experiments. With these BNC in and outputs on the front panel, we are completely independent of any soft and hardware, and you can connect our devices to all available data acquisition systems that are on the market. And this also includes our new digital amplifier, which also has these BNC in and outputs for universal connection. Our devices are a little different from other devices. For example, we have a special capacitance compensation circuitry in our amplifiers that makes them faster than others and allows fast pulses for applications, for example. And we build miniaturized head stages, which allow intracellular recordings even in freely moving animals. One very important point is that we customize our devices. And since we build everything in-house, it's no problem for us to change, for example, filter frequencies or amplification factors or current ranges of amplifiers. We build prototypes, and some of these prototypes even get products like this one, for example. It started as a prototype for Professor Bert Sackmann in Munich and became now one of our most selling amplifiers, the ELC03XS. We also build complete setups, and my colleagues and myself have been working in scientific labs for many years, and therefore we know what works together, what is needed for what type of experiments, and so we can make sure that everything is working together. And we are therefore partnering with many different companies. This is just an incomplete list. Most of them are specialists in their field, and so is Fluicell, which built their unique BioPen single cell application system, of which we will hear more in the coming minutes. So this is it from me. Thank you very much.
So thank you very much, Yen, for your presentation. That was very interesting. Before getting to the hardcore biology and have, you know, some different topic, I will take one minute to introduce what we do at Fluicell and what Fluicell is. So maybe you can stop sharing your yeah, exactly. Thank you, Jens. So just to explain what we do at Fluicell. So Fluicell basically is a biotech company based in Mölndal, so it's near Gothenburg on the west coast of Sweden, so it's quite cool there. We are pioneers in single-cell biology, tools based on microfluidic technology, and we strive to enhance cells model. So that being said, there is a real need in the biomedical research, which can be summarized as the need to deliver high patient value solution tailored treatment strategy, or the need for faster, more relevant information sooner in the discovery process, and of course the need for better utilization of high value sample. And this is what microfluidics and specifically Fluicell helps to do to their community. So this is why we have built several single cell discovery platforms. Then we have 3 platforms at Fluicell. The first one is the Dynaflow Resolve system, which is an ion channel screening platform, so for a single cell patch clamp recording. The second one is the BioPen system, the most unique and adaptable platform we have that enable you to deliver compound to target single cell or a small group of cells that can fit in almost under almost any microscope. And the BioPixlar, it's the new baby of the family, which bring a breakthrough to the industry of bioprinting by enabling bioprinting of cells with the precision of single cells and without the need of a gel or ink to create tissue. We print cells directly in native media. But that being said, let's start by the symposium. And our first talk today will be made by Professor Carsten Hoffmann. He is the director of the Institute for Molecular Cell Biology at the Hospital University of Jena. His main work focuses on working on cell-to-cell signaling and using FRET sensors for G protein coupled receptor to investigate ligands, receptor interaction, and the following signal pathway. His talk will be to present probing receptor function with intramolecular FRET sensors. Professor Hoffmann, it's your turn to start sharing your presentation and talk. Thank you for being here.
Thank you very much for these kind words. And
yeah, I will stop sharing my, okay. Okay, that's good. Thank you.
Okay. Then I hope I will be able to set up.
It's working.
My presentation. Okay. So I'd like to thank the companies, both NPI and Fluicell, for inviting me to give this seminar and present some of our data. We have actually been in contact with Fluicell already very early on. You've just seen that Fluicell had been founded in 2012. And in 2015, we purchased the first BioPen. I think it was a very early on serial number. And Gavin back then came even to Würzburg when I was there to see what microscopes we had and paid us a site visit. And I moved only a few years ago to Jena. So I would like to start my presentation with a very general introduction of a few slides and then go into the biological background and actually the use of the BioPen. So Jena is located somewhere centrally in Germany. So this is Berlin. You find Munich down here, Nuremberg in between, and Frankfurt. So we have a long distance to travel to all of these things, but it's a nice location. We're located in a really nice valley. And you see the research campus already here. This is the main research hub of Thüringen. So we two Max Planck institutes here. We have two Fraunhofer institutes, 3 Leibniz institutes. So there's a real big emphasis on, and one of the main focus of the university is optics and photonics. And around here at this corner, you will see the headquarters of Carl Zeiss located of the microscopy company, and we collaborate with them as well. So this is my office actually, where I'm sitting right now and talking to you. And this is a research merger of the university and the university hospital to combine the strengths of biological science and medical science in one hub. When we talk about conformational sensors, why do we actually study protein-protein interaction and conformational changes of GPCRs in living cells? I've printed here, or I've shown here a graphic of the three major classes of membrane receptors that actually translate information from the outside to the inside. Among them, ion channels, G-protein coupled receptors, and enzyme-linked receptors, which most of you will know as tyrosine kinase receptors. My particular emphasis on G-protein coupled receptors, because they are a very, very important class of receptors for cellular communication. And there are more than 800 different GPCRs in your human body, so different classes, which are basically involved in physiological control of any function that you can think of, be it neurotransmission or your visual system, the reception of odorants, or anything else. Most of this is controlled by GPCRs, and they need to translate a signal from the outside to a signal to the inside. And they couple via different G proteins and can evoke intracellular signaling cascades like ricin cyclic AMP or calcium, leading to a contraction or the phosphorylation of different proteins. The reason why we're interested in this is more than 30% of all marketed drugs work through these receptors, because you can guess that when they're involved in so many different processes, that the drug companies have targeted them already to bring back pathophysiological functions to normal, and or at least to hold the disease progression. And this is really why we are studying G-protein coupled receptors. The translation of information from the outside is achieved by ligand binding, and there has to be a conformational change to translate this information on the outside to something that is readable on the inside, and then for the recruitment of downstream effectors and starting a signaling cascade. So for us in molecular cell biology or in pharmacology, where in Würzburg, where I was hosted before, this is a crucial part, because the change here in conformation dictates on what happens inside the cell, and therefore, of course, the hope is to be able to control this in a reasonable manner to maybe even select individual signaling pathways. The other reason why we study this is in combination with medicinal chemists, we are developing compounds that may actually select certain functions or block individual states of these signaling cascade. I will focus on a very early step in my presentation, that we analyzed a chemokine receptor for the effect of ligand binding in this conformational change, and then what happens with the G protein. There is, of course, a lot more to study, but I will just focus today on this initial part, and I will show you some of the details. The reason or the way we do this is by actually generating fluorescence-based sensors. And I've just shown you here one of the general schemes how you can actually label different receptors. This is a size of a G-protein coupled receptor. This is currently how many people label them with antibodies, and you can see this is a huge size. We will use fluorescent proteins and a small organic fluorescent dye to have minimal invasion on these proteins. There are alternative tags like luciferases or SnapTag and ClipTag, which you can buy from other companies. And this is just in quantum dot to emphasize the size difference here. So we would like to be as minimal invasive as possible. And therefore, I first show you CFP and YFP are the frequently used fluorophores. That's the basis of FRET. So you have to have an absorption spectra and an emission spectra. And the emission spectra of the donor needs to overlap with the acceptor spectra. Excuse me. And if that is correct and in the right distance between the two fluorophores, you will get a nice thread signal. And the distance depends, of course, on the combination of fluorophores. And there is an optimum where you actually can insert your fluorophores, because if you are around the so-called Förster radius, which gives you the half maximal signal, then you get, for a short distance change, the largest signal change, actually. So for us as people in the lab cloning those sensors, that means this part is the majority of work to optimize your sensors for function. What I do differently to most people in labeling is I do not only use the green fluorescent proteins, but I was visiting Roger Etienne's lab in 2003 and was trained on a technology which was called flash labeling. So we use a small organic fluorophore, which is called fluorescein arsenical hairpin binder. And this fluorophore can be tagged with 6 amino acids only, instead of 230 amino acids, and therefore you are less invasive. The advantage for us in this today's talk is that the signal amplitude that we obtain from receptors being cloned with these technologies is about 5-fold of what we would get with a CFP-YFP sensor. So thereby, these receptors are suitable for kinetic analysis as well as concentration response curves of ligands. With respect to the kinetics, they would not be different, but this is, of course, more difficult to analyze than such a signal because you have a higher signal-to-noise ratio. The type of microscope we use is actually in custom-built microscope where we have a camera up here and an optosplit in here, then a light source. And you can see here already the conventional perfusion system, which we use from NPI for, let's say, not so costful ligands. In this device here, you have reservoirs that take 5 milliliters of solution, so small, diffusible and commercial ligands we can use with this system. What you can see here is a small tip of 200 micrometer diameter that we use for regular application to cells, and then here the recording device on the side. And I hope this video runs. That's a video where you can see actually here CFP and here Flash. The postdoc who did the work is Armand Gutbulle in my lab. And so he perfused an adenosine receptor, and I hope this live recording works and does not kill the stream. So we can see here is an on rate and an off rate when we start perfusing a ligand, and an on rate, off rate again. So if I start the video, you might be able to see that we have an on rate coming on here. This is a video that was taken with a cell phone. So you can see the actual experiment on screen, how it's done. It's really live cell. And here Armand stops the perfusion and the ligand diffuses off again or is washed off. So I stopped the video here. I hope you could see this as it was developing. And for the sake of time, I just analyzed that, or Armand analyzed that if we do and concentration here with adenosine at different concentrations, you see for low concentrations, we get a bit of an answer. And then following theory, the on rate becomes faster, the higher the ligand concentration is, while the off rate stays constant. This was done with the regular perfusion system, not with the NPI system, because adenosine is a very cheap ligand. Then we wanted to turn to a chemokine receptor. And there we already have a problem because 1 microgram of a ligand of this chemokine receptor would cost you around 3,000 euros. That is nothing you would put in a 5 milliliter tube because you can do exactly 1 experiment and then you purchase the next milligram. So this was actually the reason why we got in contact quite early on with Fluicell and Gavin to have a look at the perfusion system that we actually heard rumors of would exist and could be used. And the reason why we're interested in this chemokine receptor is it's an important receptor in breast cancer and particularly in metastasis. And there is an antagonist now finally in clinical use because a lot of women suffer from metastasis in bone because this receptor allows these cells to migrate toward a gradient where this ligand is actually highly expressed. And doctors would normally look for liver or lung tumors or brain metastasis in kidney, because these are the organs where blood flow would limit flow and the cells might have a place to settle. Bone is not really on the list of organs where metastasis would form, but these cells migrate in there, and this is really due to this receptor, and thereby blockade and understanding this receptor is vital for us to cure that. So having said this, we actually started this project in 2015 when a PhD student joined my lab in an ITN-funded or an EU-funded network, Christina Pepina, and I will show you just a figure in a second. So this ligand binds to this receptor, and by the start of the project, the crystal structure became available. So we had seen from colleagues that inside here in this receptor, this is where the dynamic happens in the third intracellular loop. When a ligand binds on the outside, this is the place where the downstream signaling molecules will bind, and we saw you can insert something in there. So Christina analyzed these receptors and then started to clone. And so this is a photo of Christina. And what she did was she actually took this third intracellular loop and inserted at several positions a flash site that we could actually label. So this is now called 226, 228, and 229 for the site where she actually inserted the 6 amino acids. What she then did was to characterize this guy here, where we now have a sensor that would respond with a change in threat, hopefully, when we apply a ligand. So you can see the expression in cells, and you can see also that this flash labeling, if you look into it in confocal microscopy, gives you a slight staining, but you can still see that there is specific labeling. What Christina did then was to determine the threat efficiency so that we could actually get what our signal would be like. We expected this signal to be here between 4 or 5% from our previous knowledge that we had with 18 other sensors for other GPCRs that we had generated. She also, of course, did then G protein activation and in different assays to show that this receptor is about 10-fold less in signaling, but nonetheless responds well with respect to ligand binding. Now the pipette comes into play because we need to stimulate this receptor. Design of the pipette is pretty easy. You have, or I should not say pretty easy, but it's straightforward. I think it's very difficult to manufacture. But what you have is 3 openings. Through one opening, the actual liquid is applied. And through these 2 outsides, the liquid is absorbed, which leads to a defined bubble in front of your pipette. And you can engulf a single cell in that. You need this holder, and that's shown here on the other side. Since we purchased an early version for your work, you can use the first 4 vials here, and the remaining vials are for waste deposit, because of course you have to deposit your waste that you absorb again. But as I've heard, there is now another version where you have more vials for actual working. The solution, then you can use this on slices or you can use this on single cells. We use this on single cell, but it's a very small amount of solution required because it recirculates. And that's the huge advantage. Our experience is that with this volume of 35 microliters, you can work for an entire day on different experiments without needing to refill it, because it only has a few nanoliters of liquid here in front, and that's the advantage. And since you have 3 or 4 in our version, 4 different solutions, you can use one of these solutions for your buffer to maintain a resting state. And then you can add an agonist in different concentrations, or even put an agonist and an antagonist in here to do switching of different solutions at the single cell level. This is what it looks like when you have the different solutions. I will not run this for long, but you can see that you can have a green solution, yellow or red. So you can, in 1 second, it should start switching faster to show you how fast actually the solution exchange can be. But up here, now it starts. Up here, you can see the aspiration of the volume that's going in. So this is actually a video that I stole from Fluicell when Gavin was visiting us and advertising this video. The second one is just the size of the bubble that you can create. So you are flexible. You can even put in multiple cells or even single cells. And you can maintain this also very, very stable for the time of your experiment. So having shown this, I'll start now with the actual experiment. We now have our pipette here on this receptor. We have expressed these receptors in regular HEC293 cells. By a normal transfection reagent, we used F18. And then we did our staining protocol with flash and actually exposed the ligand at this small black bar here. That's the time frame where Christina applied the ligand. And you can see that there is a rapid change in the ratio. And you can see this here as well in blue and yellow going to the opposite side. So this is a real indication of threat. And then you can analyze the on rate here, which Christina has done. And the on rate is in the range of 600 milliseconds. That is perfectly fine. There is no delay or something by this perfusion in this time scale. If you go to much faster time scales of a millisecond or two, then you have to use different perfusion systems. But for our regular use, this is totally fine. And then she also quantified the off rate, and this is in range of 20 seconds. So this is a number that I would like you to remember, 600 milliseconds for the activation of these receptors. Of course, we have to do control experiments to make sure that our signal is really coming from this receptor alone. So therefore, Christina cloned two constructs which either carry the fluorophore in for flash, the binding sequence for flash, or the CFP, and co-expression does not lead to an increase upon bleaching. So this is chemical bleaching where we detach this fluorophore. You can see this here declining, but CFP is not increasing, while when you have both in the same molecule, you see the CFP fluorescence increasing, which means that all our signal really comes from a subunit in this, or from one receptor, not a dimer. Then she started to analyze G-protein receptor coupling, because this is the ultimate next step. So you have an active receptor now. Now you have coupling of the G-protein and the receptor. And you can see here again, FRET in both settings where we tag the receptor and the gamma subunit, or the receptor and the alpha subunit. And this was totally unexpected, because suddenly we see a decrease. And this is counterintuitive, because if you are provoking FRET between two partners that should interact, you should expect an increase, not a decrease. So therefore, we compared these two settings, but nonetheless, in both cases, we had a decrease, which meant that they were probably pre-coupling already in a resting state. Both of these settings gave us an activation kinetic of 1.2 seconds. So compared to ligand binding and activation of 600 milliseconds, we are a step slower of 1.2 seconds. So Christina analyzed this further to convince ourselves that the perfusion system is okay, and not by switching the different perfusion systems between our long publication list, that there was a difference here. And we used the alpha-2A receptor, which we had analyzed with an perfusion system, a different one already years ago. That was mainly Moritz Bühnemann's work, here Peter Hein et al. And so the activation kinetic, we could actually recapitulate for the receptor G-protein coupling. And you see here an increase as expected and published, and the decrease here for CXCR4. So we are sure that this is a different type of biology going on. So therefore, Christina used now a bleaching experiment where she looked at these two receptors. This is steady state. This is not with a perfusion, but she basically bleached this fluorophore. And you can see here an increase in CFP fluorescence on the G protein. So they must have been interacting before. If she puts in an antagonist of the receptor under the same circumstances and then bleaches, there is no increase in fluorescence, which means she can discouple this state by putting in an antagonist. And then a control experiment, again, the alpha-2A receptor, which we had shown before was not pre-coupling, didn't show any pre-coupling. So it's another evidence that there is some interaction between the receptor and the G protein already, but something happens within tau of 1.2 seconds in this complex upon ligand application. So the next step in your signaling cascade that I've introduced you to would be the G protein activation. And here again, we used tools that we had in the lab from Joachim Gödert from Amsterdam, where we have here GI-coupled receptors tagged within CFP and YFP-based fluorophore. And you can see here a decrease that is not coming back, and the alpha-2A receptor, again, a decrease, but in dynamic one. So that means here we have something else going on compared to the alpha-2A receptor. The main point that I need to raise is here the kinetics are in the range of 4 seconds compared to an alpha-2A receptor, which is in the range of 600 milliseconds for a tau. That's an exact match of the previously published data. So we have a gap here between the interaction of the receptor and the G protein and the activation of about 3 seconds. So we were scratching our heads to try and explain this, and here's just some plate reader assays to quantify this with respect to the ligand. So we were scratching our heads and thought, what could be the reason actually for this mismatch in time scales? Because for all other receptors, we have seen a very, very rapid switch on. Even for peptide receptors like the PTH receptor, this is much faster. So an idea came up that we might actually use a dimer, and receptors of this class can dimerize. And so Christina cloned 2 C-terminal tagged receptors with the C-terminals of CFP and YFP, and then exposed the ligand to it, and again aspirated it using this biopen from Fluicell. You can see again a very nice Fred trace here. The on rate could be analyzed, and the on rate is in the range of 1.6 seconds. So it falls in between the complex rearrangement of G protein and receptor in the interaction and the G protein activation. So this then led us to propose a model where we have actually here ligand binding, where we do not know how rapid this is because we can't use, or we can't currently visualize the ligand, but we're working on this. And here we have the activation of the receptor. This is the receptor G protein complex with an tau of about 1 second. And then this dimer rearrangement would kinetically fit immediately in between here. And then we have the activation of the G protein. So this is kind of an attractive hypothesis, which we may utilize actually for further drug development. Of course, we are collaborating also with immunologists. So they were interested in a compound which is called MIF, which they call macrophage migration inhibitor factor. And it has been proposed and shown actually by ligand binding that it also interacts with the CXCR4 receptor. So here now is the strength of our approach, because the pipette can hold 4 different solutions. So we can use MIF in one tube, CXCR12 in the other tubing, and then expose the 2 ligands on the same cell, having the same identical protein content and transfection. And it's just a single cell. And what you can see here is the structure. MIF is a non-conventional chemokine, so it kind of harbors a bit of the basic structure of chemokines, but it does not contain the motifs that actually allow you to classify this as a chemokine. Nonetheless, we did the experiments, and you can see here on the left, again, our receptor construct. If you apply MIF, we get a negative result. On the same cell line, CXCR12 leads to an increase. And here's the kinetic analysis for MIF. If we use the receptor G protein interaction assay, MIF shows an increase and CXCL12 shows a decrease. It's just the same cell line. There's nothing you can argue about being different protein contents or anything. It's just the opposite direction, and again, the kinetic analysis. And the next steps would then be this dimer rearrangement. MIF does exactly the opposite to CXCL12, and MIF doesn't activate the G protein. So we can distinguish these 2 ligands now just based on our single cell experiments using these sensors. And according to our findings, we would characterize MIF not as an activating ligand, at least with respect to G protein activation, but it binds and induces different states. And colleagues of us with whom we collaborated, have picked up on this dimer rearrangement and just published a publication in the Proceedings of the National Academy of Science, where they can actually target sub-pockets here of the binding pocket and actually selectively destroy a dimer and bring this into a monomeric state. And this is going to be quite exciting when it comes to therapeutic use. This work that I'm just presenting you was published in August, and we were also asked whether we could present a figure for the cover figure, and this is really a very nice highlight for a PhD thesis to be honored with a cover figure in such a prestigious journal. And here you can see just the presentation of how we can visualize the different kinetic steps. And of course, this is now interesting with respect to treatment options for these receptors that we might have identified an additional state of this receptor that can be targeted by small molecules, and we can actually gain control of the different states. Of course, we don't do this alone. This is my group in Jena. Most work that I've presented you was done by Cristina Pepina-Viziano and Ahmad Gutbule as the postdoc, who is now picking up on these things, and Nureddin Yusuf in our funding from the EU, who is following her on these steps. And of course, vital teams for collaboration, long-term collaborators, and my former mentor, Martin Lose, who is now in Berlin. And of course, here, people in Amsterdam and in Nottingham who were involved in this. And funding is very important. Of course, we are involved in collaborative research centers. Receptor Dynamics was a network funded by the state of Bavaria, and EU funding. Novo Nordisk was interested in funding us for a kinetic project, and of course also EU funding. More interesting and important is the current group, because they follow and are currently working on these, and there's more to come. So with this, I would like to stop my sharing, and I'm happy to take questions.
Thank you very much, Professor Hoffman, for your presentation. This was very interesting. And I have to say that you have videos that we've never seen. So I see now that Gavin is laughing, but we will definitely have to have those videos back, because they are super demonstrating on what the biopen can do. And they are really talking, and we've never seen those, so they are incredibly describing what you can do with the biopen. So maybe Gavin, you have a question, I think, right? So we can start with him, and if anyone has a question, then yeah. You are on mute, I think. So we
can't hear you.
We can't hear you, Gavin.
Can you hear me now?
Yes, we do.
Yeah, it's not liking the headset. So thank you, Professor Hoffman. It's good to see your work again. Yeah, it's been a few years since we started this journey, so it's exciting to see how things are going and things. So I had one question, and it kind of relates kind of to multiple points that you have raised during your talk, particularly around this kind of dimer formation, and as you described at the start, with the G protein activation, there's actually a cascade process which happens. It's not just a single step thing. That I was actually wondering if there's been studies, or if there is like a kind of a role of the membrane, like how the reorganization has, let's say, elements of the kind of fluidity of the membrane, or the local microenvironment of the membrane, as well as the kind of possible other like dimerization. So like if, let's say, the properties of the membrane change, does it change the way that the G protein activation works?
This is just a field that's beginning to be elucidated. Of course, there may be influences of the membrane structure. People start to appreciate membrane curvature as a certain force, and some lipids may enrich in structures that are bended inwards or outwards. And of course, receptors might feel more comfortable in certain areas of the membrane. Then there are crystal structures out which show, for instance, that cholesterol has a tendency to bind to several of these GPCRs, and that's also been studied with functional respects. Then I think with respect to dimer or influence of lipids to dimers, it's too early to say. What we currently see is that that there is no clear rule of which receptors will dimerize or not, but there is an influence of the expression level for some of the receptors, which of course, if you have more receptors in the membrane, then they have to find each other more often. But there are also several receptors which have a natural affinity to each other, and some of these heterodimers serve actually different functions than individual receptors. So this is a very interesting area, but since you can imagine, we have 800 different subtypes, and if you start to do the combinatorial of that, we need more postdocs and probably more professors to do all this stuff. And but it's a very interesting area of research. Yes.
Cool. Thank you.
Now it's me. Then thank you, Prof. Hoffman, and thank you, Gavin, for this question. If there is any other question, you can use the Q&R button on the bottom of the Zoom conversation. And I think that Dr. Loser, you have as well a question for Prof. Hoffman, right?
That's right. It might be a stupid question, a very simple one, but all the kinetics you showed, the on time was always a lot faster than the off time. And I was wondering why this is, because this does not yet involve the G protein coupling and all this what comes afterwards. It's only the receptor itself and the conformational changes. Why is there still such a big difference?
That's because, I mean, your affinity of a ligand is a product of on rate and off rate. And so there, if you have a low affinity ligand, they would dissociate much faster. And this is most of the times where we see the difference. We have also come across a few ligands where we have a different in the on rate, but affinity is really something which now with these techniques surprises us. You find ligands with a similar affinity if you do in balanced and determination at equilibrium. You would conclude that they have the same affinity, but they do have very different on and off rates. And we're just starting to look into that. We also have a project on adenosine receptors where we particularly look at allosteric modulators put on top of agonists to look at their modulation of off rate. Because for industry, this is a very interesting question. Off rate is linked to residence time, and residence time is linked to most of the times duration of drug action. And if you look into the literature for failures of drug development in early phases is you have an affinity, but you do not have an efficacy. And this is very important to keep in mind, the off rate as well. We are actually able, by taking off the ligand completely, to distinguish between the effects of rebinding and of dissociation. Because if your ligand is around in the solution, it has, of course, a certain affinity and would rebind and lead to an falsification of your off rate. That's another advantage of the pipette, because it really takes out this the ligand and does not leave it in. And with the other perfusion system, this AXA8, for instance, we apply the ligand and then it stays in the bath solution. So it would accumulate over time. And we had to pay also some experience on that for very, very high affinity ligands in the nanomolar range, where we did a miscalculation and applied way too high concentrations, and then the experiment stopped after one cell.
Okay. Yeah, thank you very much.
Okay, thank you very much. So we have another question from Jana Kirsch. So do you see any mechanical disturbance of the cell due to the solution stream within the bubble in front of the pipette, or are the cells sitting tight enough on the surface to the chamber bottom?
We have to attach the cells with polylysine. So this is something which we use to kind of flatten the cells and make them more adherent. But within this biopen, we have seen way less disturbance than with other perfusion systems, because it's simply an amount of liquid that you would let the cells pass. And in this sense, the Fluicell is way more gentle than other perfusion systems. You just have to calculate easily if we, like the perfusion system that I've shown, where we have the 5 milliliter perfusion, you may go through 0.5 milliliter in 1 or 2 minutes, and just the flow rate would be much faster. So sometimes with the old system, we even lost cells because they were not attached good enough. And with the BioPen, we've never seen a loss of cells.
Okay, thank you very much. I think that we don't have other questions. I will let maybe a few seconds more if anyone has any other question. In any case, thank you very much, Dr. Hoffman. I'm sure that you have raised a lot of interest down there, and people, sometimes are shy and will email us afterwards. So if we have any inquiries, we will definitely send it to you.
Yes, I can answer that perfectly suitable. Yeah, just forward them.
Yes. So now I will share again my presentation. So I think that you can see it now. And now we will pass on to another speech today, led by Dr. Matija Rožnik, who is the Chief Sales Officer at Fluicell. And it will be about microfluidics systems for targeting individual cells. Matija, it's your turn to talk. I will stop sharing my presentation.
Thank you, Edward. So I will make, do you see my screen? Okay, so in my talk today, I really want to go into more of the systems that we have. So, kind of highlight some of the technical capabilities, as well as some of the applications that might not have been mentioned today. I have to say that Professor Hoffman did a very good job and cut a couple of my slides, yeah. So, and in general, as we already know, we are a microfluidics company in Sweden, and we are really focused on the flow of controlling the flow of liquids in small volumes. So here on the left, you see kind of our chip designs. So this is kind of a chip PDMS based for BioPen, as well as for BioPixlar bioprinter, and a glass chip for Dynaflow. So Edward already showed the portfolio of our products. As I mentioned, I just want to quickly go through each of them to kind of highlight the technical features and yeah, show some of the maybe other applications. Yeah, as I mentioned, Professor Hoffman did a fantastic job about introducing the BioPen as a system. I don't want to add too much to that introduction, other than show maybe from a different perspective how the BioPen works. So here we have kind of a mathematical model of that imaging of that bubble, right? So as mentioned, the BioPen has 3 channels at the end. It has a middle channel for which the solution is pushed out, and the 2 channels next to it are kind of a vacuum channels that are absorbing the solution back into the system. And this creates a microenvironment from where there is basically no dissolution into the surrounding areas. So one of the concepts you see here, you can really encapsulate a single cell in that microenvironment. It's very flexible. So the one of the main points of the BioPen is really the control of what you want to target when. So the timing, also mentioned in the previous talk, is you can have short pulses or really prolonged solution delivery to a particular cell. And since you have multiple channels, what is that you deliver at what time. From the application areas, it's a very broad application area that we have for BioPen. Really just some of the studies In single cells, obviously, that's one of the main application areas that we have. In this case, studying single cells, or more specifically the retraction of individual the retraction of the individual cells of a cell protrusion. So the biopen was kind of used to really target a protrusion and then induce a retraction in the cell protrusion. Another area also going to the effects of how the biopen works and is designed is cell-to-cell molecular transport. In this case, targeting a single cell in a network and then basically studying the transport between the cells, so in intercellular transport. The same concept can, for example, be used in neurosciences, where you can target a single cell in a network and observe network-wide effects. A slide that was shown by Professor Hoffman, and actually the study published by Professor Jedermark, who will go on next. Here, I just want to add that the biopen is also very, from this, it's very adaptable in a way that it can work with additional probes together. So, for example, in electrophysiology, the biopen can work with a with the pipette together to kind of deliver the solution in combination with electrophysiology recording. And another application, the new one, a bit more exotic, in mosquito neurobiology, a group from Rockefeller University studying mosquitoes. They used biopen to deliver whole blood to the tip of the mosquito blood feeding needle. And they were interested in recording the neuronal responses in mosquito after delivering the blood. And last, in the biopen part, it's another area that was mentioned. So the biopen also collects from the environment. So not only the delivery, but the biopen can also collect kind of interesting debris. But in this case, this was a surface adhered vesicles, where they basically use the biopen to actually collect these vesicles inside the kind of waste channels or specific channels, and then extracted this collection and further analyze these lipid vesicles. So much I wanted to add about biopen. Shortly, I also want to show about the biopixlar. This will definitely cut into some of the Gavin's talk, but I really have just 2 slides. Here, what we have done is really, the point is we have expanded the concept of biopen by just delivering the solutions in a very precise way to actually deliver cell suspensions through the same principles that apply in biopen. So with recirculation flow, we can now have cell suspension, deliver cells in a very precise way down to a single cell level into the defined area, into the in vitro cell dish. Some of the capabilities of bioprinting. So we really can go from placing a single cell into where you want it to position in the dish, to different combinations of multicellular 2D models up to a 3D modeling. The biopixlar technology is really for high resolution printing, where you want something printed with very precise, in a very precise way, and basically down to a single cell level. And last but not least, another compound delivery system, Dynaflow, specifically designed for ion channel screening. So in difference to biopen, this has a much wider, a much more solution delivery capability. So it has a 16 channel, 16 solutions. It has 16 channel. It is connected, as you see here, with a patch clamp, manual patch clamp. And it can also switch much faster between different channels. Another image here. So it's a glass chip with 16 solutions. Similarly to what biopen, it has a very low compound consumption. It has a very fast solution rate, because what happens is that you place a cell next to 16 channels that are flowing out in a laminar flow, and then the stage switches between each channels. And I think this is something that will also be described in more details with Professor Jedermark. And yeah, just an image how this looks like in under the microscope. So you see here is a cell on a patch pipette placed directly in front of one of the 16 channels. So the idea is that what happens is that you have laminar flow from all of the 16 channels. So the cell in this area is always surrounded from the fluid from the specific channel. And the solution exchange happens by simply switching the chip to the next channel. So the cell stays always at this particular place, and the chip moves to another channel. So this enables very fast solution exchange between channels, and is specifically for electrophysiology applications. And with that, I really want to finish my talk here. So as I said, the point for me was really to kind of show all the capabilities of the systems. And yeah, I'm open for a couple of questions, or otherwise give the floor to our next presenter.
Thank you very much, Mattia. Is there any question from? Yes, there is one. Perfect. So the question is the flow unidirectional? So I don't know if you are talking about the Dynaflow, the BioPen. Okay, so are you talking, so Dr. Padmanabhan, are you talking about the BioPen? Okay, so with the BioPen, is the flow unidirectional?
Sorry, can you specify in what is unidirectional for you?
He will be he must type.
Yeah, I see. So you showed there. Yes, there in BioPen there is a recirculation fluid. So the flow is it goes from one channel out and basically into the two channels next to it back in.
So I'll if I may
jump in. Sure, Gavin.
So yes, it's essentially unidirectional. By recirculating, what they are describing is the fluid exits one channel and enters another channel, but the same fluid is not reused. It essentially only ever goes one way. So from the delivery channel it goes out, and from the aspiration channels it goes in. But so effectively you generate what's called a hydrodynamically confined zone, where a liquid is always moving and being delivered. But the solution which is collected back isn't re-delivered. That collects back into a separate internal waste within the device, so that you don't contaminate any other cells in your sample.
So okay, so the second question is what are the advantages of the BioPen versus competitors? So
I think the main advantages is what already Professor Hoffman described. It's really targeting very precisely inside the in vitro cell culture, right? So it's targeting single cells or small group of cells, very small areas, without kind of interfering with other cells in that area. And another thing that is also very important is a low level of consumption of compounds that is being used, because what we are talking about here, it's and flow in nanoliters per second, and as mentioned, right, the maximum the maximum volume, it's actually 35 microliters. So I think these are the two main points for the biopen.
Okay, thank you, Mattia. We have another question. I'm sorry. Could biopen be used for cartilage tissue repair? So I think there must be a confusion. Maybe it's the BioPixlar, right, that we are talking.
Yeah, I mean, it depends on the application, right? So it depends what is the exact application. In general, if it's a cell delivery to something, it would be the BioPixlar, of course, right? And also with that, we would need to discuss in more specific what exactly would you want to do with this particular application. So what exactly is to be delivered and how, basically. And we are happy to take this discussion after the meeting. So I think it's in general it's very hard to answer.
Okay, thank you, Mattia. So we will connect with Dr. Dudic afterwards. And there is another question then. Do you know anything about the effect of turbulent flow on mechanosensitive ion channels endogenously expressed in test systems?
Gavin. No, because we had this discussion yesterday. So
we have had this discussion recently. So it's actually very difficult to look at effectively the shear stress which is imparted on cells attached to a surface. I mean, there's actually very many components to it. I mean, one thing that we do have is that the hydrodynamically confined flow is actually within a laminar kind of stream environment as opposed to a turbulent flow, where we're not at the kind of size scale or the flow rate where you would actually get turbulence. We're in a laminar stream. And what that actually kind of leads to is that the flow which is actually being exposed to the cell, the last couple of microns, actually where the cell is sitting, is actually a very small cross section to the fluid flow. And as Professor Hoffman was stating when they were describing their experience on the solution exchange, it's such a small volumetric flow rate, which actually the cell, or the cross section of the cell feels, that the simulated stress on the cell is on the order of about 8 piconewtons. Now, we've tried to stress activate certain ion channels to see if we can actually experimentally measure what stress will be done. So we've run experiments on astrocytes and things, other cells which actually do show stresses upon local fluid exchange, and we haven't successfully been able to determine what our actual shearing is, but we haven't been able to stress activate any ion channels to date. Now, we don't try to purposefully do that, so I imagine if we sit right over the top and directly flow all of the flow over one cell, that we probably can start to increase the shearing. But as far as inadvertently activating mechanosensitive channels, we haven't been able to do it, and no one of our customers that does ion channel work has described any, let's say, negative effects of the flow on the cells.
Okay, thank you very much, Gavin, for this answer. I hope that answer your question, Dr. Mule. Okay, so now if there is there any other question, or everyone is good? I one pop up right now. Okay, so we have a new one from Dr. Kuckley. When you bring the biopen close to the cell in cell culture or in the slice, how do you prevent that bath solution / culture medium enters into the pen by capillary forces?
I can answer quickly. So what we actually do want the bath solution to enter the biopen. That's just to clarify, we do want that to occur. We're actually introducing one, let's say, liquid compound into another liquid environment. So we're just overcoming any diffusion by maintaining this hydrodynamically confined flow region. So by continuously flowing in a laminar environment, they don't mix, but you can introduce one into another. The bath solution, as you accurately call it, is taken up into the device, but that goes into a waste region of the device. The solutions that you're trying to deliver and the solutions that are in the bath never, let's say, mix. They all waste goes to internal waste wells. So effectively, you have two wells in the chip, which would then mostly contain your bath solution, because that's what's being aspirated mainly from those two side channels.
Okay, thank you very much, Gavin. Is there any more question from any of the attendees, participants right now? Or can we maybe switch to the next talk with Professor Kent Jardemark? I will share my screen right now. I think there is no more question.
Okay.
Just to introduce briefly you, Professor,
Yes.
I have prepared a small slide. So Professor Kent Jardemark today is talking from Karolinska Institute. He's working at the Department of Physiology and Pharmacology, and he's head of the Translational Pharmacology Group. He will talk about novel antibodies for treatment of pain. Thank you very much, Kent, for joining us today. We are so pleased and so happy to see you. It's always a pleasure. So yeah, it's for you time to talk and time to amazing us with your novelties.
All right, thank you very much, Edward, and thank you very much to all Fluicell for this invitation to give this talk here about our research. It is very exciting. So I will try to share my slides here. And I hope you will see it here now.
Yes, we do. Thank you. You can go in full screen, and that will be.
See if I can use a pen here as well. Yeah, so you see my slides here. Okay, I will just bring down all of.
Yes, we see everything. Thank you.
So, thank you very much for this nice introduction. And I think the purpose with this presentation is to put your technologies produced by Fluicell in a context, how we use them actually in our, you can say, research based on what we do here in our pharmacological department. So in this case, we will talk about antibody treatment of pain or novel antibodies produced by Oblique Therapeutics, also in Gothenburg. And I will just start to give a short presentation of my group here. So as Edward said, I am head of the section of translation pharmacology, and we are in Biomedicum here. You see this fancy building here, which is constructed based on Marriott Hotel, actually, with 10 levels. So we have moved in here quite recently, 2018. And the purpose, we have 11 group members, mostly senior group members, researchers, but also a PhD student, Oskar Ljungholm, and another student. So the purpose with our research is in general to study mechanism of actions underlying pharmacological treatment of different diseases, for example, pain and cancer. I've also been working a lot with neuropsychiatric diseases like schizophrenia and depression, and have investigated antipsychotic and antidepressant drugs. We have several collaboration, and as I mentioned, we collaborate with Oblique Therapeutics. They are producing, I will talk about their platform for producing these antibodies that we are studying. We also are collaborating with the Royal Institute of Technology here in Stockholm, and SLU in Uppsala, and of course Fluicell. So I have local collaborations, for example, with Professor Camilla Svensson. She's in charge with the laboratory. They are focusing on pain as well for treatment of rheumatoid arthritis. So we are collaborating with several associates here. So before we start to talk about the specific project, then I will just give a background concerning antibody therapeutics. So this field is growing rapidly, much due to the clinical success seen in with several monoclonal antibody therapies. So for example, you know about Herceptin, targeting the human epidermal growth factor receptor 2 on breast cancer cells. That's been very successful. We also have Humira, which is a TNF receptor antagonist, and it's mainly used for arthritis diseases. And then other examples are Avastin for cancers and eye disease, and alirocumab, which is a cholesterol-lowering monoclonal antibody treatment strategy. Also, which is very interesting, is that due to advances in protein engineering, a wide variety of engineered antibody fragments have been also derived. For example, Fab fragments, nanobodies, and especially bispecific fragments binding to two antigens which are, for example, expressed on the tumor cells as well as on a T cell. So you can actually bring the immune system very close to tumor cells in this way. So from a pharmacological point of view, this field of antibody therapeutics hold promises of high specificity and affinity compared to the old strategies with small molecules, which are quite unspecific and causes severe side effects. It's also possible to have a deep tissue penetration with these antibodies, and they are quite, from a kinetic point of view, they are quite high. They have a high stability, and there are less toxics compared to, for example, old chemotherapies of cancer. So there are a lot of beneficial, you can say, properties of these antibodies or antibody therapeutics. So now I will go to this specific platform or technology which has been developed by Oblique Therapeutics. So as you see here in the title, is a novel lipid-based protein immobilization microfluidic technology by Oblique Therapeutics. And the purpose is that this technology identifies promising epitopes or peptide sequences on membrane-bound and membrane-associated proteins by using limited proteolysis. And I will come back to that in the next slide, what it means. So the strategy here is to gain detailed knowledge about the surface which is exposed, and this surface exposed functional active epitopes or peptide sequences within a native protein. And a key thing here is that the proteins that are native, so it's possible to produce antibodies that are likely to affect the function of the protein. So this will facilitate the development of efficient antibodies and decrease the need for elaborate screening procedures by lowering the amount of antibody candidates. So here is the principle of this ABIPROT platform. So the key thing here is that they are using proteases to probe, you can say, to probe the antibody binding site on a protein based on size similarity. So a protease has the similar size as the Fab fragment of an antibody. So a possible method to evaluate the surface topology of a protein is to restrict the activity of a protease to digest only the most flexible and exposed parts of a protein to which an antibody can bind to. So this is achieved in a very, very sophisticated control of the activity of this protease. That is, for example, to use low temperature, low concentrations, and short digestion times. So technically, as you see here in B here, it's possible then to immobilize the protein within a microfluidic channel, which will permit multiple rounds of this proteolysis under very controlled kinetics. And here you see here a combination of different kinetically controlled proteolysis strategies is possible then to release peptides for analysis by mass spectrometry, for example, to identify native surfaces, native epitopes or peptide sequences. Then from these peptides is then possible to produce antibodies, molecular antibodies, which can then be both agonistic and antagonistic antibodies. So now we return to the specific project here. So this is just a general description of this platform. So we have this, we are focusing much on pain, and this is why I want to bring up this project, because to describe the influence and the use of Fluicell's techniques in this, which has been very, very valuable in this project. So you may know that 20% of the world population suffers from chronic pain. And this is expected to increase with increasing aging of population. So from an economical point of view, then chronic pain has an enormous impact on economic productivity. So the price tag in the United States is $6 billion, which is more actually than cancer and heart disease combined. And indeed, there's a great unmet medical need. So only 25% of the patients receive sufficient pain relief from current treatment options. And the current pain medication, for example, the opioids, such as, for example, morphine, it has its limitation because it can induce drug dependence. It is very risky to use these kind of medications. There are quite few targeting which are ion channels here. Many of the treatments options are, for example, non-steroid anti-inflammatory compounds. So here we will focus on this ion channel or protein called TRPV1, or transient receptor potential valenoid receptor 1, which is an ion channel expressed in, you can say, primary afferent located in DRG neurons, and they are afferent, they are exposed in the dorsal horn of the spinal cord. So this channel is very, very important in mediating pain. And as you see here to the left here, it's a multimodality receptor. It can be activated by several, in general, activators such as capsaicin, endogenous ligands like NADA, protons, heat, and so on. So it's a multimodal receptor. But this has been a problem. I mean, previous efforts has been to develop small molecules for inhibiting this receptor in a clinical perspective to inhibit pain. But these molecules, they are bound to the whole receptors and blocked all of these modalities. Capsaicin-induced currents and proton-induced currents and so on. So this has caused, in phase 1 studies of patients here, it has been noticed that blockage of the heat sensitivity of this channel has resulted in hypothermia. And it has been reactions then in the phase 1 studies. So the purpose here with the project here is to just induce a kind of selective blockage of the analgesia part here, which can be induced by capsaicin, for example. So the purpose has been to develop a motility selective antibody. That is a selective antibody that retains just heat sensitivity in the channel but block the other modalities. So here's the title of the project that we have performed. So Oblique Therapeutics in Gothenburg, they have produced molecular antibodies against the TRPV1 channel, and we have investigated them by use of our models. So the purpose has been to test the pharmacological activity and analgesic effect of these anti-TRPV1 antibodies by using mainly two types of in vitro techniques, like the in vitro calcium imaging and the electrophysiological patch clamp recording. So if we start with the first one, calcium imaging, and now you have heard a lot about this very, very nice and fancy biopen. I don't think I have to go through all the details concerning using this biopen. But basically in this project, or in this method, we have used CHO cell, Chinese hamster ovary cells, and they have been able to overexpress human TRPV1 because it's very important to address here or investigate antibodies against the human TRPV1 here. And here we have used a calcium imaging technique, which is based on that we have loaded the cell with Fluicell, which is a sensor for calcium, for example, both calcium influx from outside the cell and also from, you can say, internal stores like endoplasmatic reticulum into the cell. It has been used a lot in, for example, muscle research and so on. And then we use that in combination with confocal microscopy. So here, as you see in the setup here, we can combine this, you can say, limited perfusion volume, where we just can, you can say, administer our drugs with a heat probe laser. So here, by using this heat probe laser, we can activate just a few cells in this culture here, and then see how the antibodies are affecting this, you can say, activation by heat. We have a complement here also. We have to, I mean, we were supposed to screen for like 14, 20 antibodies. So we need also a high throughput screening, a plate reader. And we have also used a calcium imaging technique here. So here we have been able to measuring the effects of several concentrations of the different antibodies in this plate reader. And here's the second method. It's related to patch clamp. And here we have used the same cells as in the calcium imaging experiments, the CHO cells expressing the human TRPV1. And here, as you see here, the patch clamp is about to, you can say, attach a cell to a pipette tip. And this, you create, you can say, a giga seal between the cell and the pipette tip. And then you have further suction here, and you will disrupt the membrane, so you have access to the internal part of the cell by the electrode solution here, normally consisting of a high concentration of potassium. And then you can position these cells in front of the tubes here, the Dynaflow tube. Maybe I've talked about this before. And it's a very effective model here. And the beneficial things here with this that it was mentioned in the previous talk is that you can perform quite fast experiments. I mean, this patching here of this cell here is very sensitive. And the patching holds for example, for 20 minutes or something like that. You see also, you can also prepare a lot of solutions here, and it means that you can do very, very effective and fast concentration response curves by using this Dynaflow technique. And also another thing there, perhaps you have mentioned that, is that the flow from the tubes here is laminar, so you have very low mixing of the different, you can say, buffers or drugs here, which is very beneficial. So you can get very nice responses from the different drugs that you're applying to this patch clamp cell. So we have added capsaicin, NADA, and together with antibodies. And here you see one example of the trace here. With NADA alone, then you can activate an inward current here, which is mediated by calcium. And then you add this antibody here, OTV4, together with NADA, and you see that OTV4 is blocking the response by NADA. And then you can, by flushing away OTV4 with this very effective system, you can retain the response from NADA alone here in the next response here. So here are a kind of summary of the results here. So first we tested like 14 polyclonal antibodies on these human TRPV1 induced responses. And we used two actually small molecules as controls here. It's the MAVA-TREP binding to, in general, to TRPV1, and this AMG 517. And you can see that they are quite effective in both blocking the capsaicin-induced currents as well as the heat-induced responses in these cells, which are TRPV1 mediated. But, which is very interesting here, is that the antibodies produced a selective inhibition of just the capsaicin-induced activation of a TRPV1 channel, but what was ineffective to actually block the heat activation of these channels. And this is on human TRPV1. So we did the same experiments in mice, on mice DRG neurons, these so-called pain neurons, and then we were unable to see any effect of these antibodies on mouse TRPV1, so to say. So here are more results. Then we selected a couple of antibodies from the previous studies here with the polyclonal antibodies, and they produced monoclonal antibodies. So here we can also see the modality that they are also modality selected. So first here we see binding studies here, that one of them, OTAB1, is have a very great affinity in binding to the show cells expressing these TRPV1 channels. But if we do not express the channel in the show cells, we cannot see this effective binding. And here we also see that O2AB1 is very effective in blocking capsaicin-induced currents here. And while MAVA-TREP, of course, and AMG 517 also are effective in blocking these currents. But we didn't get any inhibition of the heat. So as I said before, also these monoclonal antibodies that were very effective, they were not effective in blocking the heat, while these control molecules also were effective. And then we went again to made concentration response curves here. So we went on with this OTAB1 and could see that we had a concentration response relationship here. If we increased the concentration here, we saw a better effect on the capsaicin inhibition. And this was repeated by NMI in Austria. They also did the same experiments here and could see that capsaicin could be blocked, capsaicin-induced currents could be blocked by this OTAB1. So in summary, we can here then demonstrate so to say modality selective monoclonal antibodies for example, this OTAB1, which is targeting the human TRPV1. This is the first pharmacologically active monoclonal antibody targeting this ion channel, as well as the first rationally designed antibodies against an ion channel. Thus, by using this novel platform for antibody production, then Oblique has developed monoclonal antibodies targeting this human TRPV1, which is a very important target that has failed, as I said, using small molecules approaches. So due to this complexity of its activation mechanism, most compounds targeting TRPV1 have produced adverse side effects, such as hyperthermia or loss of heat sensation. So a modality-selective monoclonal antibody inhibiting capsaicin activation, but not heat activation, could then, as we think, offer a safe solution to improve the treatment of pain. So thanks to all these technologies that we have been using, we have been able actually in detail to study the effects of these antibodies on a very, very important target here, the TRPV1 channel. Thank you very much.
Thank you, Professor Jadmark. So is there any question first from any of the speakers to start? And maybe we can start with any of the participants.
Shall I take away this the slides?
Yes, you can. Or you can. Yes, absolutely.
Yeah, so I can ask one thing. So,
yes.
Yeah. So with their TRPV1, assuming that the mouse and the human one are fairly similar.
Yes.
So if I'm not mistaken, the capsaicin receptor is an internalized receptor, and the proton one is there's different locations for essentially where the binding parts are. Is that correct, right? So
yeah, right.
So
but they are, I know what you're thinking about. Yes, yes. Mm-hmm.
Yeah. So I mean, are there like different, let's say with the binding of the antibody fragments, are they only externally, binding, or do you have to also look at the transport mechanics of the antibodies to the binding sites to, activate or deactivate their receptors?
Yeah, that's very interesting. I mean, maybe you know about the previous experiments in Uvorva's lab that they have Karina and Trukulia and Uwe, they have used internal antibodies for binding the internal binding sites, so to say. But here we believe also that there are actually external binding sites for capsaicin as well, because we have a very quite fast response here. And also, which is very important, is that the antibodies, they cannot, they cannot enter the cell in this setup.
Okay, cool.
Yeah.
Thanks.
But of course, you can have, it's very complex. So you can have conformational changes by adding capsaicin in different ways. I mean, allosteric modulation and so on. We try to investigate that, the mechanisms here whereby antibodies are blocking.
Okay, thank you very much, Professor.
Yeah.
Is there any other questions? So right now we don't have questions, but maybe people are shy and some question will come up
yes.
in a few minutes. So I have so we have one more talk today. It's ex-professor, and now we have to say doctor. Sorry, he's the CTO. So he was a professor at Chalmers University before starting in 2012 Fluicell AB and becoming the CTO of the company. Now we call him Dr. Gavin Jeffries. He's an expert in microfluidics, and his topic today will be about patterning tissue for cell microenvironment engineering. So Gavin, it's your turn to talk. Make us dream now.
Okay, so thank you, Edward. So what I'm going to do is I will have the fun presentation. The last one's always the fun one. It's the quick wrap up where we get to actually look at some fancy technology and especially leave the kind of symposium with some food for thought. So hopefully I can do that for you today. So as Edward said, essentially what we're going to discuss is patenting tissues for actually cell microenvironment engineering. But while I will take one step to acknowledge Professor Hoffman's very excellent introduction to this type of technology. The whole field is generally known as hydrodynamically confined flows, where we can control the liquid environment external to the microfluidic device. And what that really empowers you to do is deliver compounds, as we've seen a few times. But also one thing I will show in a few slides is the ability to deliver things which are within the flow. So we can actually use it to bioprint cells and actually build the microenvironment that we want to probe with our single cell experiments. So I actually just grabbed this slide from an old presentation just to give Professor Hoffman an updated set of images on things. So we can actually show, how the hydrodynamically confined environment, changes, in real time. So we can see these very strongly fluorescent compounds actually changing. What we have here, or as in these two zones, especially on the right side, is demonstrating one liquid being introduced into another one, and there's no mixing and there's no loss. However, when we are dealing with these hydrodynamically confined flows, we want some sort of validation that, okay, if we're going to introduce liquid A into liquid B, we don't get lost to the surrounding area. This is very important when you're using agonist antagonists or doing any sort of electrophysiological readings. You want to have confidence in your platform. So one of the things we equally looked at recently for another reason was to actually set up a continuous recirculation zone, which is shown in the bottom, and then look at different distances away from the tip. And then by probing these different distances, we're able to actually see if any of that compound actually enters that. So we use very long exposure time on the cameras to actually see, does any leakage occur? And what we're happy to say is that yes, we actually do maintain a confined zone at 10-second continuous exposures, even 30-second continuous exposures. What we see is we go back down to background every time. We don't have any transient solutions being lost into the environment, and we can do this repeatedly. What you also get from this is by studying the hydrodynamically confined zone, we can see that we're actually getting fairly square wave activation. We get very sharp on and off times. And when you're looking at compound delivery for studying these kind of things that have been described today, looking at ion channel responses, it's very good to be able to decouple the liquid delivery from the actual response of the ion channels or the biology itself. So having these on-off times as square wave-like as possible allows you to have some sort of a decoupling of the biology from the liquid delivery as a whole. One particular example, because I have to show something different that has been seen before, is actually a project that was done a few years ago, actually studying communication within muscle fibers, actually looking at what sort of interconnectivity there are within mitochondrial networks within one particular fiber. And here we can see that we're delivering to one part of the fiber, and it's not spreading throughout, and we're not losing anything to activate larger parts of the fiber. But what I've shown so far is exactly what has been kind of raised by other people, which is this approach is very good at delivering liquid to a small area. It does so in a contamination-free manner. It does so with very little volume. And it does so with a very high repeatability, both on the area being exposed and also the timing of what's being exposed. However, this can be expanded. So as I was elucidating at the start, if we can control the liquid handling within one small region, can we use this laminar nature to actually deliver things other than solutions? So can we put something in there, such as cells, to actually micro-pattern the environment that we want to study if we're interested in how particular cell networks work, or if we're interested in what the change of the local environment is on the response of a particular signal or a response for a particular cell. Can we build that microenvironment using this approach? So we developed essentially a single cell bioprinter where we can use the power of microfluidics to position not only the printhead, but the things which it contains, in this case, it being cells. This can work in 2D and 3D, but one of the big separating factors of this technique, particularly relevant for electrophysiology or other probing techniques, is that this can be done in the liquid environment. We don't need to have anything covering the cells. In other words, as soon as the cells sit down and are happy and maybe stretching out, they can then be interrogated directly. You can use external probes. They can be connected with patch pipettes, for example. So this is very amenable to other electrophysiological and probing techniques. And like Professor Hoffman was indicating earlier, the volume usage at these sort of size scales is incredibly small. So it's very good for working with very scarce or very valuable cell samples. So just to give a little patterning example, just to show kind of what this kind of control can empower you to do, is here's just a typical pathological skin slice, just to show how cells can be patterned within a sample. And we're going to take that image, and then we're going to pattern cells in exactly the same way. So here's a little video of us essentially building that type of patterning, whereby we try to replicate the gap regions, the curvature which would be there in the histology. We try to replicate the local environment, the density of cells which are in the different areas, so that we can try to establish a system which better resembles what the cell might feel in a native environment. So what we can do is take a blueprint, it being either from a histology sample, or we can take a blueprint from something which is in research, or in fact something which is just of interest, i.e. a particular ratio of cell type A to cell type B. This can be then built, printed, and then it can be studied in however we want to. The one advantage, as I was mentioning, is that because we use no binding matrix, we can put cells on top of other cells very, very locally. And we can do this in both 2 dimensions and 3 dimensions. The kind of reason or rationale for that is if you need to really control the microenvironment. So if we need to really eloquently control the microenvironment, here being an example of a study for studying stem cell and cancer cell interactions, these are actually done on top of other cells, like seeding layers of cells, where we can pattern very precisely the different regions, so we know what the kind of contact area between different types of cells are, and we know effectively the ratio difference between the cells, so they each are in their own microenvironment. This can be done with many different things, as you can imagine. Whatever we can load into the fluidics, we can print and we can pattern. Sometimes it's more interesting to work in 2D models. Sometimes it's 3D models, as we can see in the top, both with skin. On the right side, there's a malignant melanoma model. But sometimes it's more interesting to actually just control the local environment. So we've done projects and work with people on actually patterning the local microenvironments of stem cells, these being MSC stem cells. And on the bottom right, we can equally see printing and patterning and actually allowing primary neurons to form processes, to form connections to other cells. So these can all be essentially studied so that you then have a patterned system that you can then interrogate using other electrophysiological or fluorescence techniques. This allows you and then gives you the control to build your system, as opposed to just allowing the cells to be where they are. So just as a kind of quick wrap up, what essentially HCF or hydrodynamically controlled tools allow is this ability to deliver compounds and control the microenvironment around these individual cells. This control of microenvironment can in fact be building cells around the cell that you're actually of interest in, which allows you to then build some form of microtissue from the bottom up, from individual cells. Allowing you to be able to do this means that you can get repeatable environments. So you can actually replicate the environment that you are most interested in from a single cell perspective, giving you this ability to have microorganization of the tissues, to study cell-to-cell interactions and how that actually changes or has impact on the measurement system, how maybe critical cell densities or maybe the inclusion of rare cells into the sample can affect the responses of various cellular systems. And also because we still are founded on this solution exchange, we can then go to these tissue systems and directly control the microenvironment using the solution exposure system. So, and with that, I will wrap up and I will stop sharing and I will be open for any questions.
Thank you very much, Gavin. This presentation is amazing. I mean, for everyone that work with single cells and want to create tissue model and disease model and any type of pattern. Is there any questions from our speakers? Feel free to ask. Yes.
Yeah, I would be interested in how shear forces would affect the viability of cells when being printed. I mean, it surely depends on the size of the cells, because the size of the channel the cells go through is constant.
Yeah.
So
yeah,
what do you know about this?
Yeah, so it definitely, I mean, it's always a concern. I mean, I would with regards to, and I can show if or I can make available the cell types, you know, that we have worked with. We don't notice any negative effect on the viability or the survivability. So we recently published in scientific Nature Scientific Reports using this device to actually do bioprinting constructs in 2D and 3D, and actually looking at basic like liver model systems, and whereby we actually, for all the systems we did, we measured actually the viability compared to standard cell culture and handling. And what we actually saw is that it's either identical or, you know, approximately better. I mean, because it kind of positively selects for the healthy cells because the healthiest ones stick better than the not healthy ones. So you actually can, the cells which would be within the circulation that maybe not as healthy as they should, don't stick as efficiently. So then you get the most kind of bang for your buck, if you want to say, when you're actually printing and patterning the cells. But from our kind of data, we're in the above 90%, and in some cases 95%, both viability and survivability, which has been checked.
Thank you.
Yes, Professor Hoffman, you can talk.
How long does it approximately take someone to train on this machine to really use it efficiently?
It's a very good question. I mean, one thing which we wanted to do from the start was taking actually experience from feedback from people like yourself that actually have used the BioPen, is to remove as much fluidic understanding as necessary as possible and actually switch it to just biology. So we actually use everything on a gamepad. We use a little joystick controller, and you just move around. I mean, you can use the kind of CAD type interface and you can draw what you want, or you can just run it in free run mode and actually just translate where you want. I would say that The only, let's say, hurdle to printing is being able to position, a device. It does have a series of different automated, like, focusing and everything else, like a lot of platforms do, but you still need to have some understanding of positioning a probe. But with regards to training somebody, I mean, within half a day, I mean, you can be printing fairly comfortably. But as I'm sure you're aware, handling of the cells and the biology, that's where the difficulty comes, not the actual operation of the machine.
Okay, thank you, Gavin, and thank you, Professor Hoffman, for the question. Is there any participant that would like to ask a question regarding any of the talk that happened today or the talk that Gavin just gave? It's now. Okay, maybe not. So I will share my last slide. And we are heading toward the end of this mini symposium. And first, I wanted to thanks Professor Hoffman, Professor Kent-Mark, Mattia, Gavin, and of course, Dr. Jens Loser and NPI for helping us organizing this symposium. I would like also to highlight the help of all of our distributors, AXT, that are in charge of the Australian and New Zealand area, Swift Analytical, who is in charge of United Kingdom, NBT, who is in charge of Israel, and Media Analytica, who is in charge of India. Thank you very much to all of you for advertising this event. Thank you to all the participants for your time and for, I think we have a question actually. Do you suffer any issues related to sedimentation?
Yes, I can answer. It's a good question. I mean, we do load individual cells into it. So I mean, cells will settle as time goes on. Normally, when we're suspending, if it's a small cell, then we want to try to get whatever we're patterning back into an incubator environment within half an hour-ish, so that we don't cause any kind of issues with the cells. We try to do it within half an hour. And within that kind of time scale, there's not much sedimentation. If we're working with, let's say, a model cell line, like a CHO cell, for example, which can survive a little bit longer, more like 40 minutes, 45 minutes, then typically what we will do is resuspend within the well after maybe 20 minutes, half an hour, just to make sure that it doesn't, the concentration's approximately, equal. But there is a little bit of sedimentation, but during the standard printing time, we don't notice that much effect. And also, I should note that we can, and one of the things we've tested is we can add a concentration of PEG to the solution to change the viscosity slightly to minimize sedimentation if it appears to be, an issue. Tested viability in those regards too.
Thank you very much, Gavin, for this. So I think that we answered all the questions. So let me get back to the end of this mini symposium. Thank you very much for all your time, for being participant, for asking all those questions. Please, at the end of this symposium, you will have a small pool with 2 or 3 questions. Take it. It's very important for us to know what you have learned, but also to know if we can keep in touch, because we have very interesting things that we always come up with, and we don't forget to always mention that we are from academia. So we keep that spirit in mind and in line with what we are doing with the scientific community. And we really love to share all that knowledge with you. So don't hesitate to contact us, and please take this poll at the end. Thank you very much again. And I hope that we see you soon in another mini symposium. Thank you, professors, and thank you, Jens, and thank you, Gavin. Bye-bye.
Bye-bye. Thank you very much.